two populations within a species are locally adapted to different habitats. So in this hypothetical example, a southern population has higher fitness in its native habitat in the south, and a northern population has higher fitness in its native habitat. So we see that neither population can be optimally adapted to both habitats. And I'm really interested in this question of reciprocal local adaptation or fitness trade-offs. So thinking about how common are trade-offs is a really interesting question. So this meta-analysis by Hereford in 2009 reviewed reciprocal transplant studies to see how often populations had fitness advantages when they were native and disadvantages when they were non-native. And those points are indicated in that upper left quadrant. So we see trade-offs were the most common pattern observed, but only 45% of the data points fell in this quadrant. So it's interesting to think about why less than half of the time we actually observe fitness trade-offs. So that brings me to a big question I'm interested in, which is when does adaptation to one habitat result in fitness trade-offs in an alternate habitat? And I think, so we all know that selective agents can vary across years. So in that study, the Hereford meta-analysis, only 9 out of the 74 studies actually measured local adaptation across more than 1 year, and that was 68 populations. So looking just at those populations where their fitness was measured in more than 1 year, we see that about a third of them weren't consistent. So a third of those populations were not always locally adapted or not locally adapted. So the results were not always consistent across years. So I think it's really important to keep in mind that measuring local adaptation in 1 year might not represent what's often going on if selective agents vary. Also, I think about trade-offs at different levels. So there can be trade-offs at the level of the genotype, like in the examples I just showed, where a southern genotype does better in the southern habitat. But obviously underlying those genotypes are adaptive traits, and underlying those traits are genes. And we could see a trade-off at the level of the genotype without necessarily seeing trade-offs at the levels of traits or genes underlying those traits. And I'm interested in exploring trade-offs across these levels. So my study system is Leptocypin parviflorus. It's an annual wildflower native to California. It's in the phlox family. It's self-incompatible, and as you can see, there's variation in its flower color. So I study 2 populations that are adapted to 2 different soil types, one of which is serpentine soil. Serpentine soil is a really interesting habitat to study adaptation because it's a really harsh environment for plants. It has poor water holding capacity, low calcium to magnesium ratios, low levels of essential nutrients, and high concentrations of heavy metals. So looking at the distribution of serpentine soil in North America, a lot of it is out on the west coast. And my study site is indicated by that red star. I work at Jasper Ridge Biological Preserve. And at my study site, I study 2 populations that are located in very close proximity to one another. So the pink circle is where if you can see that, is where a serpentine population grows on serpentine soil, and less than 100 meters away, that white circle indicates the location of a population growing on sandstone soil. I measured soil moisture across the growing season in both of these habitats, and it's interesting because I find that the soil moisture dries out more quickly and is consistently drier on serpentine soil than sandstone soil. And this is consistent with what we expect, that serpentine soil typically has low water holding capacity. So thinking about the phenology of this species, they germinate in December and January during the rainy period, and they establish as seedlings in early February. And so this is a really critical period for them for survival in terms of how much rain occurs during that time. And they start flowering right towards the end of February or early March. Basically, once the soil starts drying up, that typically triggers flowering time, and they have to set seed before the soil gets too dry at the end of the spring. So the populations I study also exhibit this Watercolor Polymorphism, where the serpentine population has pink flowers and the sandstone population has white flowers. And this is a heritable Mendelian trait. I also see gene flow occurring between these populations. And so evidence for this gene flow came from prior work in our lab that used AFLP markers to look at FST, which overall was 0.12. They also measured trait variation in both of these populations, and of all the traits measured, found flower color and flowering time were very, very divergent between the two populations, especially relative to that FST value. So that indicates that those traits might be involved in local adaptation. So my study questions are, first, are these populations locally adapted despite their close proximity and ongoing gene flow? Do adaptive trade-offs vary across years? And what traits contribute to fitness trade-offs, and what is their genetic basis? So to answer the first question, I did a set of reciprocal transplant studies in 4 years, where I took both populations and planted them in both soil types in the field. So I'm going to show you my overall results across the 4 years. These are the least square means using year as a covariate, and I also use Astor transform data, which is Astor is a way that I can combine multiple fitness components like survival, number of flowers, number of fruits, and despite the fact that they have different distributions. So across the 4 years of study, I see evidence of local adaptation where the sandstone population does better in its native habitat, the serpentine population does better in its native habitat. So I do see evidence of local adaptation, but I'm interested in understanding whether these trade-offs might vary across years. So again, these are combined fitness components, but these are for each year that I did the study. And I see evidence of local adaptation in reciprocal local adaptation in 3 of the 4 years. I circled the one year on sandstone where I don't see any significant differences between the parents. So it's kind of interesting to think what selective agents are varying across the years that might be contributing to differences in these patterns. And when I did this study, there was a drought period going on in California, so I was really interested in the role of soil moisture in mediating fitness differences. So to understand this, I did a greenhouse experiment where I grew plants on both soil types in the greenhouse, and I had 2 treatments. One where I watered throughout the experiment, and another where I just stopped watering once the first plant started flowering, and then to roughly mimic the natural phenology in the field. And here are my results. So looking first at sandstone soil, I see that the sandstone population has higher fitness in the constant water treatment. But in the drought treatment, that difference is actually totally reversed, where the serpentine population actually outperforms the sandstone on sandstone soil in a drought treatment. On serpentine soil, it looks like the serpentine population does better in both treatments, but that difference is not significant with constant water. So clearly the soil moisture interacts with the soil type. I also wanted to see whether these patterns would show up in the field. So I did some manipulations in the field where I had plots on each soil type where I had a control treatment that was unmanipulated, supplemental watering treatment, and a weeding treatment. And I did 2 replicates for each treatment on each soil type. And here are the results for that. So looking again first at the sandstone soil on the left, I see the sandstone population outperforms in the unmanipulated treatment. But when I add water, that difference, that fitness advantage is actually increased. So that, that kind of supports what I saw in the greenhouse study. And interestingly, in the weeded treatment, I actually see no difference between the populations. And I think this maybe provides some insight into mechanisms underlying adaptive trade-offs in these populations. So potentially, being adapted to serpentine soil might result in a trade-off in competitive ability. And the presence of competitors on sandstone might be limiting the serpentine genotype's ability to invade those other habitats. Looking at serpentine soil, I see the serpentine population outperforms the sandstone no matter what the treatment is. So it does appear that adaptive trade-offs do vary across years, and it appears that soil moisture seems to be mediating these patterns. And finally, I'm interested in what traits contribute to fitness trade-offs and what is their genetic basis. So I'll put this slide up again to remind you that flower color and flowering time are highly divergent among the populations. So the serpentine population has pink flowers and it flowers earlier than the sandstone population. And both of those traits are heritable, so in a common environment they exhibit those traits. So thinking first about flower color, it's interesting to consider why it varies among these populations. And so some possibilities are that it's under selection by pollinators. It's also possible that the gene for flower color is tightly linked to another gene that affects fitness, or potentially the gene underlying flower color is pleiotropic and has multiple functions. So first I asked whether flower color was under selection by pollinators, and I did studies, observational studies in the field, looking at pollinator visitation. Both populations are visited by bee flies, and in my studies I saw that the pollinators exhibited no flower color preference and no constancy. They visited flower color morphs at random. So it doesn't appear that flower color is under selection by pollinators, which is what we typically think is driving flower color variation. So I think the two possibilities are these two. However, thinking about linkage, this species is self-incompatible, and there's evidence that there's a lot of gene flow occurring. So if flower color is linked to another gene that's important for adaptation, I would have expected that recombination would have broken that association apart by now, unless it's in a region of the genome where there's no recombination occurring. So I'm exploring what the role of flower color is in local adaptation. So one possibility is that it's related to drought tolerance. So a study on a species in a related genus, Linanthus perryi, found that color was implicated in drought tolerance here. So here pollinators, there's blue and white flowers, and pollinators do not have flower color preferences. Instead, the blue flowers did better in drought years, and the white flowers did better in non-drought years. So potentially a similar mechanism is going on in my system. So I've been doing a series of back crosses where I've been trying to introgress that pink flower color locus into the white sandstone genetic background. And I put these out in the field in 2014. So here I'm going to show, so the one problem with this is I don't know the genotype of the plant if it dies before flowering. So right now I can only measure the pleiotropy and fitness on plants that survive to flower with these lines. And I'm working on making better, so near isogenic lines that are homozygous, and so that I know the genotype before flowering. But on sandstone soil where there's high survival, I can look at relationships between this genomic region and traits or fitness. And so here I'm showing flowering time, and I have the flowering time of the sandstone population, the flowering time of the serpentine population, and then these back cross lines that basically differ only at one locus, only at the flower color locus. And I had two independent genotypes of these lines. And so it's really interesting that I see a significant difference in flowering time between pink morphs and white morphs, even though they're the same genotype at they only differ at that one genomic location. And that difference represents 36% of the difference between the serpentine and sandstone parents. So potentially I'm interested in further exploring this and trying to understand whether it's flowering time that's under selection, or maybe flowering time is also pleiotropic and related to drought tolerance, which other studies have shown. It might also be related to some aspect of competitive ability. So flowering later might allow the sandstone population to invest in more vegetative tissue and outcompete competitors. So I'm still exploring that. The anthocyanin biochemical pathway provides a lot of candidate genes for me to study further too. So I'm interested in investigating the underlying genes underlying that. And with that, I would like to thank my help and thank you for coming.
Anyone has one quick question? Maybe we can get it in.
Yeah.
So the weeding treatment had a really dramatic effect on fitness of the serpentine genotypes in the serpentine.
Yeah.
So why would there be a selection for a loss of competitive ability where interspecific competition seems to have the most dramatic effect on fitness?
Yeah, that's a really good question. I was really stumped by that pattern as well. I was not